Advanced subsidiary levels, your syllabus code would be 9700. We're starting off with chapter 1, which is about cellular and O cell structure. And under that, 1.1 would be microscopy. I would like to start off by saying that there are two common types of known microscopes. There are more variants, but there are two common types of microscopes. And do not worry about my handwriting, it's going to get better. I need to warm up a little bit, a tad bit, that's why. So first of all, you have seen this in your everyday life. This is what you call a light microscope. Not because it's light and not heavy, and so simply because it uses light to observe specimen. And then there's a much more advanced form of a microscope. This is typically referred to as being an electron microscope. It's much larger than the light microscope. This uses electron beams to visualize specimen. When it comes to the light microscope, it's pretty straightforward. We have one type, we have this type and this type only. But when it comes to the electron microscope, I can actually give you two variants of it. When it comes to the world of electron microscopes, we have a transmission and scanning form difference. Okay, that's too much. Let me clarify. The first core type of electron microscope is known as a TEM. TEM stands for transmission. Transmission electron microscope. I'm not going to write that. Simply transmission electron microscope. So what's so special about this? The electron beams. So electron microscopes obviously use electron beams, right? When it comes to transmission electron microscopes, the electron beam is passed through. That looks like beam. Beam is passed through the specimen. That's the thing. It is transmitted through the specimen. Hence the name. It's transmitted through the specimen. The specimen being observed. And then there's the second variant known as an SEM. This stands for scanning. Scanning electron microscope. So just like the name suggests, the electron beam would simply scan a surface. For example, if this was some form of specimen and this was the electron beam, if it's transmission electron micrography, it's going to go through. It's transmitted and then observed. But if it's the scanning variant, only this electron beam is just going to hit this, scan the surface, reflect back, and then give us a reading. It's very simple, very straightforward. That's simply the difference. Now, when it comes to these two microscopes, you have to know their distinctive differences. I'll be talking about a specific feature, and that specific feature as part of the light microscope and the electron microscope. I'm gonna have to move this here. Oh my god. Okay, I'll place it here. Right. So the first feature I'm going to talk about would be the radiation source. Simply, we're talking about light, so we're using visible light. It has to be plugged in. That's how you get the light from the illuminator. And then when it comes to the electron microscope, obviously we'll be using electrons. Now the wavelength, when it comes to the wavelength, you know what you mean by the wavelength of something. Simply when it comes to visible light, we are talking about the wavelength range of 400 all the way to 700 nanometers, like so. And here's the deal. Here's where most students actually mess things up. Here, when it comes to the wavelength of electrons, for your levels, you can refer to it like this. You simply refer to the wavelength of electrons as being roughly 1.2 nanometers. I'm going to give 1.23 just for specificity, but roughly refer to it as being 1.2 nanometers. That would suffice. Don't go overboard. Yes, there are different wavelengths according to the pulses and the pulse systems used by these electron microscopes. There's a huge variant of all of these things, but don't worry about any of that. Simply refer to this numerical value and that should suffice. Now there's something known as the resolution in the microscopic community. I'll be talking about this after I complete this little chart sort of thing, but for now just remember resolution is 1/2 of the wavelength. Meaning if you divide this by 2 you get the resolution. If you divide this by 2 you simply get the resolution. So using that little bit of knowledge, I'll be extrapolating that a little bit more, don't worry, but for now simply we'll divide this by 2. So roughly I'll just like take 400, and so typically we say that the resolution, it has a specific name, a specific definition, I'll be talking about that, but simply 1/2 of the wavelength. So here that would be 200 nanometers. Now here when it comes to the resolution of electron microscopes, you typically refer to them as being 0.5 nanometers. Okay, now if you refer to this, actually it's going to be 0.6, I know, but it's a range again. Just refer to it as being 0.5 nanometers. Okay, not 0.05, not 0.005, simply 0.5 nanometers. Okay, so technically speaking, if we compare light and electron microscopes, they give us a much better picture. That's the idea. I'll be touching upon that idea a little later. Now, when we talk about what type of specimen we can actually place, it is not an issue if you place an alive specimen. But when it comes to the electron microscope, you can do that. The specimen has to be both dead and dehydrated as well. We cannot have any form of moisture. And there's a difference when it comes to the staining methods that we use. For light microscopes, I'm pretty sure if you have been under any form of biological perspective, you know, we can use simple colored dyes. Bromophenol blue, methylene blue, hematoxylin and eosine, and yeah, that's about the number of stains that come to my mind. There's a lot more. But when it comes to electron microscopy, we can do that. It's actually more expensive. We have to use things like heavy metals, mercury, cadmium, those stuff. Those are the staining things that we can use, right? It's a simple comparison of both of those things. Now onwards to specific terms that you have to be aware of. Let me pick a nice color. What color did we not use? Okay, that's interesting. Right. The first term would be magnification, nothing new. Magnification. How would you define magnification? If it was given to me, this is how I would define magnification. The number of times an image is large. An image is larger when compared to the object, to the size of the object, simply. The object to the size of the object. How would you remember the magnification equation? Now, I'm pretty sure you've used triangles or different other methods or some other forms of memorization, but throughout all these years, up until, I don't know, like 5-6 years, this is the best way I have found, and this is the way that I tell most of my students to follow. It's simply I am, you are, I am. Image size is equivalent to the actual size multiplied by the magnification. 2 small tips I'll give you when it comes to magnification is, first of all, you have to be mindful of units. you have to be very mindful of units. That's the first thing. Okay. And then secondly, apply properly. What does that mean? Now, when you're moving things around, you can mess things up a little bit. But if you experience that will almost never happen. So if I were to rearrange this equation for magnification, m would be simply I over a. Because this a goes down, I over a would be equal to m. Don't mess that around. If you rush or if you don't have that much experience to recategorize equations, that's going to be a problem. So do practice that. Just be cautious of that. That's my point. Right. We will be attempting a lot of questions and practice papers on magnification specifically, but for the sake of this part, I'd like to give you a small, simple sample question that I'm going to build right now. So if you were given that the actual size of an object was 5 millimeters, the image was drawn up until 550 centimeters, calculate m. Simply calculate m. So I'm going to put myself in your shoes and we'll see how I'm going to attempt this. So first of all, I'd write down the equation. I'll remember that I equals am and I'll write that down. And then I know I want magnification. So I'll rearrange things. M is equal to I divided by a. So that's what happens. Now, what is the size of the image? Always write things down. That's something I can tell you because examiners always give you marks for your working. Please write it down if it's an MCQ, of course. You won't have space and they're not really checking that. They're only checking the answer. But if it's a theory or practical based thing, always write down your steps. It's one of the most important things I have to keep repeating. Okay? Right. Now, m equals I over a. I is 50 centimeters. Right. So let's write that down. What about the actual size? That is 5 millimeters. So then you do 50 over 5. You get a value. You write that down. Completely wrong. Why? What's the issue? Refer to this. You have to be mindful of units. Whenever you're dividing, this is not even magnification knowledge. This is basic mathematics knowledge. Simply, if you're dividing two quantities, both units have to be equal, right? There are cases where that's not applicable as well, but that's the general rule. So what do you want to do? You want to convert centimeters to millimeters, or you want to convert millimeters to centimeters? I'm pretty sure the most simple thing would be to convert centimeters to millimeters. You simply multiply by 10. So when that happens, let me keep it as is and then multiply. So 10. So 50 centimeters would be 500 millimeters equivalently. And then we have 5 millimeters. 5 millimeters. 500 over 5 would be simply 100. So your magnification would be 100. Most of the time a question that I get is, what's the unit for magnification? Now do you see when you do this, millimeters to millimeters cancel. 500 over 5 would be 100. Do you see? You're left with nothing. Magnification does not have a unit. It's just into 100, into 200, into 400. That's the point. Magnification has no units. So that is how you would simply apply this equation to calculate magnification. There's a ton of practice questions available on my website. Please do go and attempt them. You do have mark schemes as well. And we are going to discuss a lot of these questions coming up as well, especially with regards to MCQs and passing MCQs and a little bit of exam tactics also when it comes to this. Now let's move on to the second term. This is a quite new term for you. Now we're going to be talking about resolution. Now, if you're into photography or videography or something like that, resolution is not a new thing to you. Simply, it describes the clarity of an image. The higher the resolution, the better things are. The more clear things would be. This is basically the same idea when it comes to magnification and microscopes. So I'm going to word it like this. See if you can get the gist of it. Now, resolution is the ability to distinguish between two distinct points. Two distinct points of any specimen. Let me explain that a little bit using a small visual. So if there's this point here and this point here, now you can clearly distinguish between them, right? That is there and this is here. Yes, but what if this was the case? Now they're very close, but you still can distinguish things like that, but it's much closer now. So you can simply see that resolution is going down. The idea of resolution is going down. Clear distinction, clarity reduces. That's the idea. Don't think of it too much. It's not that important. So the general rule would be if the magnification goes up significantly. So if magnification increases, our resolution factor actually decreases. That's the idea. We call that an inverse relationship. The more we can zoom in, you have experienced this in your day-to-day life as well. The more you zoom into a photograph, the hazier it becomes, right? The less clear it becomes. That means resolution is going down. Resolution is going down. Now that half business, do you remember I told you this is this half, blah, blah, blah. So let's talk about that here. Now the deal with that is simply, typically, resolution is half. It is half the wavelength of whatever radiation we are using. 1/2 the wavelength of the radiation used. For example, I'll use the exact same things. If we are using visible light, the smallest wavelength that we will get, or lambda in this case, would be 400 nanometers. To obtain the resolution, you just simply divide it by 2. Therefore, resolution 400 divided by 2 is simply 200 nanometers. 200 nanometers. In the same fashion, if you divide the wavelength of electrons by 2, you're going to get the resolution as well. Now onwards to a concept that is going to be totally new to you. We're talking about eyepiece graticules and stage micrometers. Now that if that sounds like gibberish, probably is. Let me write things down. So this is what we'll be doing. We are going to be calculating the size of an object, of a specimen, of anything, object, using something known as an eyepiece graticule. So that's how this is pronounced and written. Eyepiece graticule. And we'll be using something known as a stage micrometer. If both terms are very, very new to you, that's okay. Hold on. I'm going to show you how we're going to be attempting this. Now, in the world of microscopy, everything's tiny now. So on the stage, now the microscope has a stage. Now this microscope has specific parts to it as well. I'm not going into that much detail yet. You don't have to know about that yet. When it comes to that, we'll be talking about that in the practical aspect, the practical part, not yet. So when it comes to the stage, we typically place something known as a micrometer on that stage. That's why we refer to it as a stage micrometer. Whenever we place the stage micrometer on that, it has these fine lines like divisions. Now that made no sense, I know. So I'm going to bring in a simple image that is going to help you visualize what the eyepiece graticule does and what a stage micrometer is and how exactly it's going to help us distinguish and provide a proper size for our specimen. Right. So first of all, pay attention to this. These are the two distinct parts. Here we have the eyepiece graticule, and then here we have the stage micrometer. It is not complex, but it's tricky when you first see it. If you're not properly introduced to it, that's going to be a problem. Now here you can see this has a lot of divisions, this thing, and here we have some large divisions on this thing. Now what do all of these things do? What do all of these things represent? You don't have to know in theory. You don't have to know any theory regarding this at all. All you have to know is how to do this. So with that, we'll see. Now, this is the simplest logic. I'm going to give you a small formula equation. It's not really a formula. It's not really equation. This is something that is going to help you answer any form of this question, and I'll apply this right here. I will apply this right here and show you how the application is going to help you as well. Let me pick a good color. Okay, that's good. Right. So this is how the logic flows. Okay. Whatever size we have. So the representing size. Now this is not going to make sense in one go, obviously. So the representing size over the number of graticule units multiplied by the number of, I'm going to use GU for graticule units, number of graticule units of size. Okay, that made no sense. I know, that's okay. Now we'll see how exactly we are going to deal with this. This is the logic, this is the eyepiece graticule, and this is the stage micrometer. The best way I have found to teach this, to show this, to go for this, is to simply go and refer to a question. I'm going to bring up a question right here, and we will be going through this and we'll try to clarify and complete this right. So a student is viewing some onion cells under a microscope. The image below shows the cells with an eyepiece graticule on the left and a stage micrometer on the right. If the two things confuse you, it should not. The eyepiece graticule has a lot of units, so meaning small divisions like this. The stage micrometer does not have that many units at all. Now note that each large division on the stage micrometer here is 100 micrometers. So let's label that. So this is what they're saying. Now this is a large, this is a large, this is a large division. So each large division is 100 micrometers, meaning, so if this was 0, this would be 100 micrometers. That's the idea. This would be 200 micrometers. That's simply the idea. If that is 100, that's 200, and it's going from increments in 100. Each small division is 10, so that means this is 10, 20, 30, like so. No issues. Now let's see what they're asking for. Use the stage micrometer to calibrate the eyepiece graticule and calculate the actual length of the cell labeled CD in the image. Now first of all, let's refer and look at the image. I want to zoom in. Now do you see? The more I zoom in, the less clear it becomes. What's being affected? Resolution is being affected. That's the idea. Okay, let's see where we have cell CD. So they're simply referring to this cell. Okay, that's the cell they're referring to. So cell C starts from here basically and ends here. If you just see what that length is, how many graticule units do we have up to here? So here it's 25, 30, 35, 40, 45, 50. So that here is how many graticule units, how many GUs? 25 to 50. That would be 25 graticule units. So the length of cell CD is 25 graticule units. That we know. Now we have to see how many graticule units represent a specific length. Meaning now if I refer from here all the way up until here, how many graticule units is that? So we're starting with 20, 30, that's 10 there, that's 10 there, that's 10 there, and that's 10 there. That's typically the case. Let me show you. Why is my control key not working? Oh my god. Zoom out, please. Zoom out, please. What? Okay. Now see, typically eyepiece graticules are known for incrementing by 10 by 10 by 10. That's the idea. Okay, so here, how many 10s? 10, 20, 30, 40. So here we have a total of 40 graticule units. Now, if we are going to refer to this, if we're going to refer to this little equation, let me replicate that and bring it down. Make it a little smaller. Okay. Now you don't actually want this thing as well. You don't really want an equation as well. That was just to give you a way. When I first figured this out on my own, I didn't use any form of equations. I just used pure logic. That's how I did it. Now here you can see this is the idea. Number of graticule units would be 40. Now see here we have 40. First we have to get the scale. How much is how much, and then refer that to this diagram. So 40 graticule units are representing a size of, what's the size here? 100 micrometers. So then, now we have the scale, all good. Now you have to go to this and see how much this much is, how much this much is, this length, this much. So here we only have 25 graticule units. So you simply multiply by 25 graticule units. When you go for this, so 10, 100, 0, 0, cut it out, and then you're going to get 662, right? Let me double check. So that's 100 over 40 into 25. Yes, that's basically 62.5 micrometers. So cell CD is 62.5 micrometers long. Typically for graticule units and eyepiece graticule calculations and all this stuff, you typically have a range. So your mark scheme range would look something like 60 to 65 micrometers. So if your answer is well within the range, you will absolutely get marks. You don't have to worry about that. Because now if you really zoom in, you kind of can see that this might extend a little bit more. So there's like 2 more graticule units, so that you can take it as 27 graticule units and not 25. But you don't have to be that precise. You have to know the logic of working this out and how to obtain a graticule unit measurement. That would be all. Simply speaking, the more you practice this, the better the logic is always going to come to you, and it's not hard at all. If this did not go into your head, try this question on your own, replay this back, and you'll see. With that, that is all I have to say for 1.1 microscopy.